Good afternoon. We have your exams for you. You can get them after. Was that a sigh of something? What was the average? 76. And based on the results, we've decided to make the final not cumulative, but just from now on, okay? Yeah. It was my idea. Saul didn't want to do it. Just kidding. We agreed. All right, so everybody got that, including you people not here in class. The final will not be cumulative. It'll just be from the lecture after the midterm. Okay? Today we are going to talk about a particular virus which is unusual for this course. We usually don't have lectures on individual viruses. In fact, I designed this course on purpose not to do that, rather to do it in terms of function and steps of replication. But this is a really important virus. This is the pandemic of our era, so it really deserves a lecture of its own, and that's, of course, HIV and the AIDS pandemic. And this story began in 1981. This is actually a reproduction of the original Morbidity and Mortality Weekly Report, a publication of the Centers for Disease Control and Prevention. And here is the beginning of that article. In the period October 1980 to May 1981, 5 young men, all active homosexuals, were treated for biopsy-confirmed pneumocystis carinii pneumonia at 3 different hospitals in LA. 2 of the patients died. All 5 patients had laboratory-confirmed previous or current CMV infection and candidal mucosal infection. So this was the first report of this unusual cluster of opportunistic infections. And it was followed by many more. And I remember very vividly coming to Columbia in 1982 and sitting down with colleagues, and they're saying, "Have you heard about this new infection? No one knows what it is." Well, shortly afterward, it was sorted out. And it's turned out that this infection was caused by a new retrovirus, human immunodeficiency virus. That wasn't the name originally given to it. It was given 2 different names, and eventually at some point the nomenclature committees got together and came up with this. So the virus was first isolated in 1983 from the lymph node of a patient with lymphadenopathy, swollen lymph nodes. And this is one of the typical presentations of this infection. This happened in Paris. It was subsequently isolated at the NIH and UCSF. There's a lot of controversy about who discovered this initially. Books have been written about it, but it's clearly first isolated in Paris. Electron microscopy of the virus and sequence analysis showed that they are lentiviruses, which was at this time already a known group in the retrovirus family, which we'll talk about in a moment. And so here is a cartoon of the virion. It's a typical retrovirus, has an envelope. There are glycoproteins in the envelope, and within it is a core with a variety of proteins, reverse transcriptase, integrase, RNase H protease, and the viral RNA, of which there are 2 copies. And here's an electron micrograph. So this has an unusual conical-shaped core. This is characteristic of the lentis, and this distinguishes it from some of the other retroviruses as well. So the retrovirus family, we have talked quite a bit about retroviruses in terms of replication, but not so much about disease. It is divided into a couple of sections, if you will. There are orthoretroviridae. There's another section which I haven't shown here. And this is further divided into alpha retroviruses, beta, gamma, and delta retroviruses, and epsilon, and then finally lentiviruses. So the known human retroviruses are either delta retroviruses, HTLV 1, 2, and 3, human T-cell leukemia viruses. These are actually discovered before HIV, and then HIV 1 and 2. These HTLVs, by the way, infect many millions of people worldwide as well, 10 to 15 million. For the most part, these infections do not cause disease, but there are rare cases of T-cell leukemias and other inflammatory diseases as well. So today we'll talk about HIV-1 largely. HIV-2 was discovered subsequently. And these are lentiviruses. So that's what I mean when we call them lentis. That's the way they're classified. Here's a nice schematic of the virus particle. This is from a very nice Swiss website called Viral Zone. They make nice pictures of viruses. So you can see again the conical shaped core, which contains the 2 copies of RNA, reverse transcriptase, integrase. This is embedded within an envelope virion. The envelope's on the outside. There is a protein, a matrix protein that gives the envelope some support. And you notice here there are only a few glycoproteins drawn, and this is accurate. These virions don't have a lot of glycoproteins embedded in them. Some of the other envelope viruses we've talked about, I show you pictures, and the outside, the envelope is packed with glycoproteins. That's not the case with HIV virions. Now, the genome, as you know, is a plus-strand RNA, and it encodes a number of proteins, which are shown here. From our previous discussions of retroviruses, you may remember that they encode the GAG protein, which is a structural precursor. It gives rise to all the proteins that are in the virion, the capsid, for example, the nucleocapsid protein, which binds to the RNA, so forth. Reverse transcriptase is encoded in the Pol region. And then we have the envelope glycoprotein, of course, which is which is embedded in the membrane of the virus. These viruses have, in addition, many other proteins. They're called accessory proteins. And you see some of them written here, VIF, TAT, and a few REV, for example. And they have a variety of functions that have been sorted out over the years. One of them is VPU right here. It's very faint. And we'll talk a little bit about that later. These all have very specific functions, and they're not found in all retroviruses. So for that reason, we call these complex retroviruses. There are 2 distinct groups of human retroviruses. We have the leukemia viruses, HTLV1, 2, 3, and 4, and the immunodeficiency viruses, which we'll talk about today, HIV1 and HIV2. Again, these are lentiviruses. When these were discovered, lentiviruses were not new. There were already a number of lentiviruses of other animals already known. For example, equine infectious anemia virus, which causes an immunodeficiency of horses. And this was isolated in the early 1900s. So the idea of an immunodeficiency causing virus wasn't new. It had been seen already in other animals. So the name, of course, is acquired immunodeficiency syndrome. And the syndrome, which we've used quite a few times already in this course, is the occurrence together of a characteristic group or pattern of symptoms. Now, usually when you call something a syndrome, it's because you don't know what causes it in part. And here we now know the cause, but syndrome has stuck with the name. But there are other afflictions that still use syndrome in the name because we don't know what causes them. HIV-1 is the etiological agent of AIDS. There is no question about this. If you search the literature, you will find many people who believe that HIV does not cause AIDS, and this is completely wrong. This is pseudoscience, wrong stuff. It's just nonsense. It's a waste of time to read it or to even think about it. So this hypothesis has been very well demonstrated in a variety of ways, and one of them is the inadvertent infection of people with HIV-contaminated blood. In the very beginning of the epidemic, people working in healthcare institutions would stick themselves, or transfusions would be given without knowing that there is HIV in the blood. And these people were infected. So it's a good fulfillment. I shouldn't say good. It is a fulfillment of Koch's postulates. You put the organism back in the host and show that it causes the original disease. So HIV causes AIDS. Don't believe anyone who tells you otherwise. In fact, there's some very respectable virologists who continue to say that HIV does not cause AIDS. But they're from California. In the U.S., HIV has killed over 500,000 people, which exceeds all the combat-related deaths in all the wars fought in the 20th century. Over 1 million people in the U.S. are infected. About a quarter of them don't even know it. They haven't been diagnosed. There are 40,000 new infections every year in the U.S., 70% men, 30% women. I'm going to give you a lot of statistics. I don't expect you to know these statistics. I'm just trying to impress on you the extent of this problem. Half of all new infections in the U.S. occur in people 25 years of age or younger. So here are some graphs to give you an idea of the pattern of AIDS in the U.S. starting from the 90s here up till 2007. Last year, a very comprehensive report was published by UNAIDS, which has all of these statistics in it. This is the estimated adult HIV prevalence, ages 15 to 49. This has been rather steady over the last years. It was initially on the increase since 1990, but it's steady. These are three different estimates. This is a high estimate, a low estimate, and the middle. The number of people living with HIV has steadily increased over the years, and the number of AIDS deaths peaked around 1995, has gone down, and depending on which curve you look at, it's either rising again or remaining constant at 20,000 a year in the US. And the global picture is shown here. These are the number of individuals with HIV infection in each continent, and then below it is the range. So we don't have terribly precise numbers, as you can see from the range. North America, 1.3 million. You can see the greatest burden is sub-Saharan Africa, 22.5 million. It's probably higher because this was a couple of years old already. So globally, there are many, many people infected, 33.2 global infections. The number of people living with HIV in 2008, here are some numbers. What's really disturbing is that children under 15 years, there are quite a few of those here, 2.1 million. People newly infected in 2008, there are in the millions for adults and 430,000 for children under 15 years. And then AIDS-related deaths in 2008. These are global numbers, again, 2 million total, of which 280,000 are children. Now, if you wonder how this fits into the pattern of diseases and infections in general, I show you this slide, which is mortality and morbidity due to selected infectious diseases. And so here's the annual number of deaths. So we'll look at this in the different infections. So lower respiratory tract infections are the biggest killers, and this includes bacterial and viral infections. Diarrheal diseases, remember Victoria Falls, 2.1 million, right? Yeah, this is all from losing water for the most part. HIV/AIDS, so is number 3, 2 million deaths, morbidity and mortality a year, followed by tuberculosis, malaria, measles. Look at measles, well up there with these diseases. Neglected diseases, and even polio is still quite high up here. So just to give you a perspective of what's going on globally, there's still quite a bit of work to do. Now, as far as controlling AIDS goes, we will talk about this a lot more when we talk about antivirals and vaccines, but I just want to mention that triple drug therapy has slowed the pandemic, that is using 3 drugs at once to get around the problem of resistance. And we'll talk about the numbers later in another lecture. But this is only good in countries with money because these are very expensive. So unfortunately, poor countries aren't able to use these triple drug therapies. So in poor countries, people continue to die because they can't access antivirals. Bill Gates could probably help out here. Now, these are some of the antiretroviral agents that have been approved in the US. The pathway of AIDS antiviral discovery has been phenomenal from the onset. There's been an amazing amount of research going into this, and you can see there are over 20 different drugs of all sorts, and we'll go into these in another lecture in more detail. These were initially used individually, but they rapidly selected for viral mutants that were resistant, and then the idea came to do them in combination. So now we have fixed dose combinations of 3. There are currently 6 different combinations of 3 different drugs that are approved for use in the US. And in particular, this pill called Atripla, it's one pill that has all 3 in it. It makes it very easy to take these drugs as opposed to taking 3 separate ones. So this is called HART, or highly active antiretroviral therapy or combination therapy. So this can really extend the lifespan of individuals that have or are infected with HIV-1. But the problem is there's no cure. We can't get rid of the virus. Remember, these are retroviruses. So you should know that these integrate their genomes into your cells. So you can't clear virus. Yes?
So when a doctor or a nurse or anyone is pricked in the hospital and there is suspicion that they might have been pricked with a needle that had HIV or AIDS on it, are these the medications that are used?
Yes.
So how
Well, if you can catch them before integration occurs to a large extent, then you can prevent them from being infected. So in the early days, in the early stages, when it's an acute infection, if you prevent enough virions from being made with the antiretrovirals, you can catch it before it goes into this stage. Yes, sir?
There's another point, and that is that the virus is actually not that infectious.
Yeah.
So that's another reason.
And we'll talk about that in a moment. So yes, if you're suspected of being stuck, you get onto antiretrovirals right away. So we have antiretrovirals, but we don't have a vaccine yet. It's a lot of work in this area, but it's a really tough problem, as you will see. So you can't block primary infection. People still get infected, they get diagnosed, and then they go for antiretroviral therapy. So by that time, it's too late. You've already got a pool of integrated proviruses in your body that you can't get rid of. As a consequence, you cannot stop taking your drugs. You have to take 3 drugs a day or whatever for the rest of your life. If you stop taking them, the virus will once again multiply within you and eventually cause disease, as we will see. The main problem is that there are reservoirs of cells, long-lived cells that remain within you. So if the cells turned over very quickly, in the face of antiviral therapy, you could get rid of all the cells. But there are some very long-lived reservoirs that exist. So there have been people on antiretrovirals for over 10 years, and they still have proviral sequences in certain pools. And we're not even sure what all these pools are. Some of them are long-lived memory T cells, but there may be other pools as well that we don't understand. And we know this because many people stop taking their drugs. They feel great after a few years and it's expensive, so they stop taking them and they get AIDS again. So these experiments happen all the time. You also get drug-resistant viruses even on triple therapy, and you just change the combination. Drugs are expensive, which is another reason why it's problematic. And all this amounts to that AIDS is becoming a third world disease. You saw the numbers in sub-Saharan Africa. They can't take care of it even with antivirals. They can't afford them, so the virus spreads without control. HIV-2 is restricted primarily to Western Africa. It's not as widely distributed. There is homology with simian immunodeficiency virus, and we'll talk about the emergence of these viruses later on. Humans who are infected with HIV-2 have antibodies that cross-react with simian immunodeficiency virus. And this virus causes an AIDS-like disease in macaques. And so it is used as a model for trying to understand HIV pathogenesis. It also doesn't cause disease in some kinds of monkeys, which also helps us to try and understand the pathogenesis. If we can understand why it can't cause disease, maybe we can get clues about intervention. And I'll tell you a little bit about that in a moment. This is a table showing the known lentiviruses, that is, viruses of this same group as HIV. You can see the horse equine infectious anemia virus and the kind of disease it causes. There are lentiviruses of sheep, goats, cows, cats, and of course our primate simian immunodeficiency viruses and HIVs. And these viruses are all related, as you would expect. They probably had a common ancestor, and studying them can give us clues about where HIV came from. Our current thinking is that both HIV 1 and 2 came from separate colonization events of simian viruses in humans. That is, 2 separate events, one for HIV-1, one for HIV-2, where the virus went from an animal into a human. And we'll talk about what kind of animals that might be. You'll see that they are involving monkeys, and in Africa, which is where HIV-1 and 2 emerged, it involved trapping monkeys and slaughtering them and selling them at market. And so the virus jumped from those animals, which had virus in them, and into people at some point. To do this, first we have to talk about different subtypes of the virus. The virus is, HIV-1 anyway, is divided into what are called clades or subtypes. These are basically antigenic groupings of the viruses. There are currently 10 different clades, A through J, and they are based on the sequence of the envelope and the GAG genes. So this is basically a sequence alignment. And there are very distinct geographical restrictions of these clades, which is why they're important to know about. So here's a map of the world which shows you where the clades are. They're color-coded here You can see, and you can see that they have very distinct restrictions. For example, in the areas with the highest rates of infection, which is typically sub-Saharan Africa, clades A, C, D, and E predominate. Most different clades are found in Central Africa. That is the most diverse numbers of clades. Elsewhere, one or two predominate. So, for example, here in the US and in Western Europe, predominantly clade B. But it does change with time. And over time in Africa, we've seen the spread of C clade, which was initially minimal there, and it has taken foothold and spread. So the ability of people to travel extensively makes it easy for these clades to move around. And that's an important consideration when you devise a vaccine, because vaccines are likely to be clade specific. And so you may make a clade B vaccine for the US, but as people with other clades move into the US, they're going to overcome that protection. And so you have to be able to be flexible. Yes?
So do the different clades correlate with virulence?
They can. In some cases, they can correlate with virulence and transmissibility. But we don't exactly understand that. So here are some sequence comparisons among these lentiviruses to give you an idea about the origins. So here is this is a phylogenetic tree where you take the sequence and you calculate the amount of homology among the sequences, and then you make a tree based on what you think might be the way these evolved from a common ancestor. And this amount of distance is 1% sequence difference on this kind of a tree. And you can see here the primate immunodeficiency viruses on the top, and the bottom are all the others, the horse, cat, bovine, goat, etc. Immunodeficiency viruses are down here. So here we have HIV-1, which is right next to a SIV, which was from a chimpanzee. So this is SIV chimpanzee. And then we have HIV-2, which is from a sooty mangabe virus. So we believe that both chimpanzee SIVs and mandrill SIVs are very closely related to HIV-1. It's likely that HIV-1 evolved from these viruses. So again, the meat slaughtering scenario that I suggested probably involved a chimpanzee or a mandrill. HIV-2 is believed to have evolved from viruses that are found in sooty mangabes, another kind of monkey found in Africa, often kept as pets. So there's close contact between people and these animals. They can scratch you and transmit the virus to you. All right, so two separate colonization events from some kind of a monkey into humans. Now, when did this happen? The oldest known sequence of HIV that we have was from 1959. It's a serum sample that had been stored from a Kinshasa male. And so this was taken for other purposes in 1959, and it was stored, which was good. And then in 1998, they went back to these. They were looking throughout any kind of clinical specimens that they had and looking for HIV by a variety of tests, PCR, for example. And it was possible to amplify the entire genome from this sequence, from this serum sample, and sequence it, which is really amazing. And so that gave us a way to clock the evolution of HIV from 1959 to the present. So that was really important to have that sequence. So that's called Zaire 59. And as far as we can tell, the sequence puts it at the ancestral node of this D group right here. So these are all different HIV and SIV isolates arranged according to group. So another way of arranging these viruses is by groups, which are collections of like sequences. So the D group shown right here, the ancestor was something related to ZR51. In just about 2 years ago, another sequence was obtained from 1960, and that's from the Democratic Republic of the Congo. This was actually from a paraffin-embedded, formaldehyde-fixed section of some tissue from someone that was in an archive. And they got this out, and they were able to pull out almost the entire HIV sequence from this paraffin-fixed sample. It's very similar to what was done with flu, if you remember. I think I talked about this. The 1918 flu was reconstructed in the same way. And that sequence is ancestral to the A group here. So in other words, ancestral means it predates all of the known sequences that we have in that particular fork. Now, what's interesting is if you compare the 59 to 60 sequences, you see a huge amount of diversity already. So this is already multiplying extensively in people. You can use that sequence to calculate a clock by which how much the virus mutates each year, and then you can back calculate to where this probably went from a monkey into humans. And that, from that kind of analysis, we suspect that the M group up here originated around 1930, and probably these viruses started in a chimpanzee and then infected people around 1930. And the N and O groups are now thought to have originated from separate transmissions from monkeys into humans. So the date 1930 isn't obviously firm, but somewhere around there is where we believe that HIV-1 went from a chimpanzee into humans. The other day someone asked me, I don't know if you've ever heard of this book called The River, which suggests that clinical trials with polio vaccine in the 1950s in Africa was the source of the HIV pandemic. You ever heard about that? No? Well, it's a book written about 8 years ago now, before all this sequence analysis was done. So the thesis was polio vaccines were tested in Africa. They were propagated in chimpanzee cells of some sort, which had HIV in them, and then they were fed to people, and that's what started the outbreak. So that's totally wrong, because we now know that these viruses, by the sequence analysis, originated in 1930. And so from 1930 until 1980, what is going on? It took that many years for this virus to replicate and spread to the extent in populations that it became a notifiable disease. And this is a graph that illustrates what we think is happening. This is the population of various cities or towns in Africa around the epicenter of where we believe HIV emerged. So this is starting in 1880. This is the population. And these are when these individual places were founded. Kinshasa, Brazzaville, Yaoundé. These were all places in the area where we believe the original transspecies transmission occurred. And you can see very, very small populations. And none of these had populations greater than 100,000 until well into the 40s and 50s. So it took a long time for the virus to spread from the initial infected individuals throughout the population. Then you see very large explosions of population. There were massive population movements. There were wars that displaced people. And then in the 1980s, all of that was enough to get the virus in many people. So it's a very interesting story, which really makes sense in terms of population growth in that country. And then, of course, once it was established in Africa, it could spread elsewhere as well. Now, there are some interesting things we find out about studying HIV replication in the animals where it came from. It turns out that old world monkeys are resistant to HIV infection. So the animals from which the virus came are resistant to it. It has now evolved in people such that it's no longer be able to infect these animals. Now, there is a protein in these monkeys called TRIM5 alpha, and that's why the cells of these animals are resistant to infection. This is called a restriction factor, and it's believed to have protected these animals from HIV infection over the years. Our TRIM5 doesn't block HIV, obviously, otherwise we wouldn't be talking about this right now. For some reason, our TRIM5 has had to stay constant. Maybe there are some other pressures put on it we don't really understand, but it doesn't have the sequence that a old world monkey TRIM5 alpha has, and therefore we are susceptible to infection. But studying this provides us with some ideas about how we could interfere with HIV infection. So that's why this kind of research is informative. Also, if you take cells from South American owl monkeys, these are also resistant to HIV infection. That's because of a different protein. It's TRIM5 fused to a second protein called cyclophilin A. Yeah?
Does TRIM5 alpha serve like another function in humans and monkeys, like the real reason why it's there? Is it just,
It's thought to be part of the innate immune system of animals. So this is the only known function at the moment. Whether it does something else, we don't really know. Old world monkeys have a similar fusion protein that also helps prevent HIV infection. So this is TRIM5, and this is how we think it works. So normally in cells that don't have TRIM5 alpha or one that doesn't block infection, the retrovirus fuses with the cell membrane. The core is deposited in the cytoplasm. It undergoes reverse transcription and eventually goes in the nucleus so that the DNA can integrate into that of the cell. If you have TRIM5 present, one model is that there's some protein, there's TRIM5 in the cell. That's the green protein here. It binds the core and prevents it from either uncoating. It may block reverse transcription. It may block transport into the nucleus. It could target it for degradation. We don't know how this works. But the idea is that this protein by itself, or maybe with another protein, somehow it blocks the integration of the viral DNA. So TRIM5 alpha defines very precise steps. People are trying to sort out how this works, and maybe you could then target it with an inhibitor of sorts. All right, let's talk a little bit about transmission, which Professor Silverstein mentioned before. It is not a particularly infectious virus, not like measles virus, which is highly contagious. HIV is not spread by respiratory, alimentary, or vector routes, some of the more common routes of transmission that we've talked about. Here are two different graphs depicting the transmission of HIV infection and by what mechanism. Here in the U.S., from 1981 to 2003, male-to-male sexual contact, which peaked around 93 and then went down, now is sort of plateaued, intravenous drug use, and heterosexual contact, which is on the rise. So in the U.S., you can see for many years, male-to-male sexual contact was the primary mode of transmission, but now all three are very close in their contribution to transmission. If you look at the rest of the world, you have a very different picture. And that's here on the right in the pie chart. 80 to 85% of transmissions of cases are transmitted heterosexually outside of the U.S. And then homosexual transmission, 5 to 10%, intravenous drug use, 5 to 10%, blood transfusions, 3 to 5%, so contaminated blood, where you don't pick up the presence of virus, and other unknown mechanisms of transmission. So this is important. HIV transmission in most of the world is by heterosexual transmission. Now, how does that happen? So you would think immediately that it's virus that is transmitted in sexual fluids, but there's still some debate over whether it's a virus or virus within cells, or maybe both. I was just reading an article this morning where they say we still don't know the relative contribution of free virus versus virus within cells. So this is a summary of various studies where they looked for virus in either cell-free fluids or infected cells. So you can see cerebrospinal fluid, ear secretions, etc., feces, milk, plasma. Plasma is a large one, and that's one of the ways you transmit it, of course, by blood transfusions. Semen also has virus in it. Vaginal cervical secretions also contain virus, but you see some of these have very little virus, like this one. Infected cells, peripheral blood mononuclear cells, these are the number of positive samples out of the total number that were tested, so 89 of 92 in this case, not much in saliva. Semen, 11 out of 28, and vaginal and cervical fluid, 7 of 16. So in these areas, there are also infected cells. So the question is whether it's virus or virus-infected cells that is transmitting the infection is still not established. Very difficult to do these kinds of studies. And that's an important question because, remember, a vaccine that induces antibodies will prevent free viruses from infecting, but it's not going to have an effect on a virus-infected cell. So you really have to figure that out before you can design an effective vaccine. So this is a study done in Uganda a number of years ago, which measured the probability of HIV transmission per coital act in monogamous heterosexual HIV discordant couples. So that means they were monogamous as far as they told everyone, although people never admit it when they're not. That's the problem with all of these studies. And they're HIV discordant, which means one partner is HIV positive and the other is not. So you can follow these couples and ask what's the probability of transmission. And so you see here, depending on the viral load, HIV RNA copies per milliliter of blood, there is a very different transmission rate. So very low viral loads, there's very low transmission. But here you can see that with 1,700 to 12,500 RNAs per milliliter, you have a probability per 10,000 coital acts 10, roughly 10 in 10,000. And higher probability if you have genital ulcer diseases. Makes it easier for the virus to get into your bloodstream, of course. And these probabilities go up with more virus in the blood. And I think now the general rule of thumb is you have about a 1 in 200 chance of contracting HIV by sexual mechanisms, okay, based on studies such as this. So it's not terribly infectious. Yes?
To do that kind of study, do they have to tell them not to be
Yeah. No. They have to tell them, you have to always advise people to have safe sex. And when you do vaccine trials, you have to do the same thing. You have to give them, all the right advice. But there will always be people that don't listen, and that's where you get the data from, unfortunately. Yeah, you're obligated on ethical grounds to do that. You have to educate them, yeah. Okay, so some of the reasons why transmission is not terribly effective in other routes is because air drying reduces infectivity, heating. So in very hot climates, it's inactivated on surfaces. It's easy to get rid of it on surfaces with bleach or alcohol or pH extremes. So not a very hardy virus. And that brings us back to structure. And here again, we have a virus which isn't very resistant, so it ends up being transmitted by routes that don't require resistance to these kinds of extremes. Okay. Now, we learned many weeks ago that the receptor for HIV requires 2 receptors to get into cells. One of them is CD4, the molecule on CD4 positive T cells. So that's shown here in green. So here's a CD4 positive T cell, and that's the receptor there, and there's another one. But the second receptor that's required, it's a chemokine receptor that's needed for infection. And remember, it's because fusion has to occur. It can be different according to the strains. There are 2 different general kinds of strains of HIV that we define within an infected individual. We have T-cell tropic strains, and these use a alpha chemokine receptor called CXCR4. And then there are macrophage tropic strains that use a beta chemokine receptor. So just 2 different co-receptors that define the 2 different strains. And I'm telling you this because we have to talk about the evolution of these strains, and so you need to know what I'm talking about. All right, so let's go through a typical course of HIV. So you get infected either by free virus or virus-containing cells, and the dendritic cells, of course, come to the infected area, and they interact with virus, which is what their job is. So you typically get infected with macrophage tropic strains, the ones that use that beta chemokine receptor that I just showed you. Okay, so not T-cell tropic. This is typically the ones that are transmitted seem to be the macrophage tropic strains. On the surface of dendritic cells is a molecule called DC sign, and this is what it stands for. Dendritic cell specific ICAM3 grabbing, that should be a G, non-integrin. Don't worry about the name. Just the fact is that the virus attaches to it. It doesn't go into the dendritic cell, though. It just attaches to it and lets the dendritic cell take it into the lymph node, which is where the CD4 positive T cells are that it wants to infect. So basically, the dendritic cell delivers HIV to where it goes. I think this was a similar situation for another virus that we talked about previously. So again, the virus doesn't get into the dendritic cell. It just attaches to DC sign on the surface. The dendritic cell, having sensed an infection, goes into the lymph node with the intent of delivering this to a T cell, and the virus is going to replicate in that T cell. So the virus then goes to the lymphoid tissues where it replicates. You get a lot of virus produced. Then the virus spreads to other areas, other lymph nodes, where it can find more CD4 cells in which to replicate. There is an initial immune response, and we'll show some graphs of this, and the immune response is effective. It downregulates the virus load. So you have initially a peak of virus production, and then we have antibodies and T cells kicking in, and they limit infection, but they can't eliminate it entirely. And after about 6 months from the primary infection, you reach what's called the viral set point, And you have that level of virus in your blood. So you have initial burst of virus, and it reaches a set point after about 6 months, at which you stay for maybe up to 10 years, depending on a variety of factors. So here's a wonderful EM. This is a dendritic cell in the lower right, and it's got HIV particles attached to it. So this dendritic cell has picked up the virus particles by DC sign, and this dendritic cell is interacting with a T cell, a CD4 positive T cell, which is what the job of the dendritic cell is, right? But inadvertently, it's delivering virus to the T cell. These are CD4 positive T cells, and the virus is now going to infect the T cell. So it's a cruel twist of evolution, I guess, that the very mechanism intended to clear pathogens is enabling infection by this one. Primary HIV infection clinical characteristics, about 50 to 90% are asymptomatic, so you don't really know that you have been infected. Symptoms, when they do occur, 5 to 30 days after exposure. What kind of symptoms? A lot of nonspecific viral symptoms, like we've talked about before, which are due to cytokines being produced. Fever, fatigue, malaise, arthralgias, headache, nausea, vomiting, diarrhea, adenopathy, swollen lymph nodes, pharyngitis, weight loss, leukopenia. Does anyone know what leukopenia is? Reduced white blood cells, leukocytes. I'm not going to ask you that, but you should know these words. How about thrombocytopenia? What's that? Low platelets. So infection can cause that as well. And you all know what your liver is. Median duration, about 2 weeks. So these are primary infection symptoms. You can't tell that you have HIV, okay? Very nonspecific. Here's a very interesting slide which shows the effect of virus replication in the colon. So this is a photograph of someone's colon, and within the colon are these collections of lymphoid tissue, GI-associated lymphoid tissue, all these lumps here. I mean, these all look like tumors, but this is normal bowel. This is the way it's supposed to look. These are full of lymphocytes. These are basically lymph nodes on the lining of your bowel. And in an individual infected with HIV, in this case, the virus was delivered into the colon. After a few weeks, all these lymph nodes are gone. The virus has destroyed all of them by replicating in CD4 positive T cells. So the bowel looks very clean, but in fact, it shouldn't be that way. So here's a graph of the course of infection. We've just talked about this initial phase. So this actually looks like an acute infection. You get infected. You have a rise in viral RNA on the bottom here. This is HIV RNA copies per mil of plasma. So you have a peak, which goes down by about 10 weeks or so. And that could just be an acute infection. On the top are some measurements of lymphocytes. So you can see here there's an initial decrease in the number of lymphocytes. So a normal person, about 1,000 CD4 positive T cells per microliter. And there's an initial drop there caused by the virus killing them. It's replicating in them. There is an increase in CD8 positive T cells. These are cytotoxic T lymphocytes whose job it is to kill virus-infected cells. So there's a burst in those. And we think those are important for controlling that initial viral level. And then the virus levels go down. So these immune responses, by the way, this top purple line is all CD8 positive T cells, and these are virus-specific CD8 positive T cells. All right, so then the virus is cleared, or not cleared, but the levels go quite far down by about 10 weeks, and then you have a long period where there's a low amount of virus, and there are really no symptoms, but during this period, the virus is killing CD4 positive T cells. The CD8s remain constant until the very end. And then when these reach a low level, now you have development of AIDS. Here, the blue shaded area. This is where your immune system can no longer repel other pathogens. And you get opportunistic infections like the pneumocystis that was in the first slide, and Candida, and many other infections as well. So the number of T cells begin to decline. As those decline, you can no longer fight off infection. The T cells are also going down. Virus RNA levels now rise significantly towards the end. This is 10 years now, 8 to 10 years. And then they drop again because there are no more CD4 positive cells for the virus to replicate in. And then eventually you get a secondary infection and you die from that. So HIV doesn't kill you, but something else kills you because you have no immune system left. It's completely destroyed it. Okay?
Till these years when when the virus is replicating, our immune system is also fighting that?
Yeah, we're making antibodies. We're going to show you what's going on there. Yeah, there's a give and take with that whole time. We make antibodies, the virus changes. You make new antibodies, the virus changes. So you never eliminate all the virus. Yeah. So if you take antiviral drugs, you can keep yourself at a very low level without going into this AIDS phase, basically. And you can live like that forever. Well, I don't know about forever, but whatever a normal lifespan is. So an established infection, which is that middle part, you have active viral replication. It's low levels, but the virus is still replicating. There are major reservoirs where the virus is replicating outside of the blood. One of them is the lymph nodes, where the virus replicates, eventually destroys all the lymph nodes, all the CD4 positive cells in the lymph nodes. Gastrointestinal tract, lympho-reticular tissues, we saw a picture of that being destroyed. There's also reservoirs in the central nervous system, the genital tract. There are many, many different kinds of viruses. They exist as a quasi-species. We'll talk about what that means in evolution, but as this virus multiplies, it mutates extensively. And you have huge populations of diverse viruses. Every day, you make 10 times 10 to the 9th virions. So during this middle phase, it's a huge amount of virions. And then they're destroyed in the same day. Huge turnover of virions, all right? And that gives you a lot of genetic diversity. That's what we mean by quasi-species. Half-life of HIV in the plasma is less than 6 hours. So a virus is made, and 6 hours later, sometimes 30 minutes later, it's gone. So very rapid turnover during that intermediate phase of disease. So this just shows you some of the various cells that are compartments for HIV replication. In the blood on the left here, we have productively infected CD4 positive lymphocytes. And these are just half-lives of these cells. So these are very short-lived cells in the blood. They account for about 99% of the virions that are made in the blood. These have a very short half-life. You see 6 hours here. And they can go on and infect fresh CD4 positive lymphocytes, which continue to be made until they're outpaced by viral destruction. There are also other compartments. There are latently infected lymphocytes in various other compartments that we mentioned, which don't make virus, but they have viral genomes in them. That's what we mean by latently infected. And these have longer half-lives. Look here, 8.5 days and 145 days for these. And these will divide and make more cells that are latently infected and have half-lives of 145 days. So that's one of the reasons why you can't get rid of the virus. These cells have DNA integrated in them, and the drugs don't do anything with that. And many of these can go back into the blood and serve to produce, they can be activated and produce virus as well. So if you stop taking antiretrovirals, these latently infected cells will move into the blood and start producing virus that will reinfect everything. Now, the course of HIV infection is variable. I have told you of a specific course, but it can differ significantly. We have typical progressors, which I've told you, an initial acute infection, clinical latency, and then many years later, AIDS. This is a reproduction of that initial graph. We also have what are called rapid progressors, where you have, again, the primary infection, and then AIDS occurs much more rapidly than in a typical progressor, within a few years rather than in 10 years. We don't understand why this happens, but people would like to know because it may give us clues about how to prevent disease. And then we have the very interesting non-progressors. These individuals are clearly infected, and yet they go on for years and years, and they never develop clinical AIDS. And there's a question mark here because many of these individuals are detected in this intermediate stage, so we don't actually know if they have had an initial acute infection. They were clearly infected at some point, but we don't know if there's a burst of RNA here. They can live forever. These are non-progressors. They're also called elite controllers because they control the infection very well. So some of the determinants of what's going to happen, whether it's fast or slow or intermediate or you don't go to disease at all, whether the virus escapes from immune responses. We make antibodies, and these react with viruses, but the virus undergoes mutations in these various proteins that allow the virus to escape. 10 times 10 to the 9th virions per day. Among all those virions, there's probably viruses resistant to every antibody that you already have. And if you make new ones, the viruses will change. So it's really a race you can't win. Sometimes viruses sustain deletions. One of those accessory proteins that I showed you on that initial slide, NEF, sometimes deletions arise in the virus, and those are not pathogenic, and that can explain some of the long-term survivors. The tropism of the virus changes. Remember, I said infection occurs with R5 virus, the macrophage tropic viruses, but in the course of an infection, by the time you have AIDS, you now have mainly X4 viruses in you, the T-cell tropic viruses. And this is often associated with increased pathogenicity and disease progression. There are also subtypes that have different potentials for transmission in different ways, and we mentioned that earlier, that some of these subtypes or groupings of antigenic types can have different pathogenicity. Now, not just CD4 cells get injured in AIDS, but many other components of your immune system as well. And I'm showing you this just to give you a sense of that. So I've told you that the virus infects CD4 positive T cells and directly kills them. The virus is cytopathic in those cells. It destroys them, and that's one of the main reasons why AIDS occurs. But the virus also destroys many other components. Look at this, CD8 positive T cells. Remember in that graph I showed you a decline in the number of CD8s, CTL, cytotoxic T lymphocytes. We think this occurs because the precursors of CD8 have CD4 on them. They may be infected by the virus, and that will end up killing the CD8 cells. Monocytes can be killed. B cells, NK cells, they can all be destroyed. Their functions can be destroyed by virus infection. In most cases, we don't know how this happens. So that's why we have a total immune collapse, because we lose many of our main components of the immune system. Here's what happens in a lymph node. This is a normal lymph node at a very early stage of infection. There's a little bit of virus. So the bottom slides here are the white dots are virus RNA being made. So initially, the dendritic cells are bringing in virions. They're transferring it to the lymphocytes that are resident in the lymph node, and then the virus is going to go out into the blood. The lymph node, upon getting dendritic cells in it that are reporting trouble, begins to proliferate. It's called hyperplasia. Make a lot of lymphocytes so that they can go out into the blood. And that's why early on in infection you have swollen lymph nodes. And that's one of the diagnostics. That's why the virus was originally called lymphadenopathy virus, because one of the symptoms was swollen lymph nodes. In the intermediate stage of infection, you have more sites of viral replication in the lymph nodes. There's what is called a follicular dendritic network in the lymph node where the dendritic cells are interfacing with lymphocytes. This begins to fall apart. And then by the end or late stage disease, there are no more lymphoid cells in these lymph nodes. There's all connective tissue now here. The virus has destroyed pretty much all of the lymphocytes, and what's left are some dendritic cells that aren't very good at trapping virus particles at all. So the whole function of the lymph node has really been destroyed by virus infection. And that's why, in part, you get immunosuppressed. There are also neurological disorders associated with AIDS. Besides immunosuppression, about 30% of infections have neurological disorders of various sorts. These are both direct and indirect. So by indirect, for example, you can have opportunistic parasitic infections, toxoplasma, cryptococcus, that give you neurological symptoms. You can have various lymphomas, tumors. You can have encephalitis, peripheral neuropathies, muscle pain, paralysis, weakness. So a variety of clinical features, most of which we don't understand the etiology of. And that's in about 30% of infections. Here's a schematic of what we think is happening in the central nervous system. So the virus, here's a virus particle here, is able to get into the CNS. Remember, a long time ago we talked about the blood-brain barrier to getting into the CNS. So these are endothelial cells. One idea is that the virus can cross endothelial cells. In the CNS, the virus can infect astrocytes. It can infect oligodendrocytes. but it does not infect, and it can also infect microglia, but it cannot infect neurons. HIV doesn't infect neurons, yet it does produce neurological symptoms. So the idea is that all these infected cells are producing a variety of cytokines and perhaps viral proteins that give the neuronal toxicity. Okay? So this is just an example of a macrophage infected, which is a microglia in the CNS. If it's infected with HIV, it produces all of these chemokines and cytokines and other metabolites. And what these have been shown to have effects in the CNS. So, for example, quinolinic acid produced by macrophages is associated with CNS disease in HIV-infected children. And cytokines, these cytokines, TGF-beta, IL-6, TNF-alpha, are associated with other CNS signs. So we think that not direct virus infection of neurons, but rather all the chemicals that are produced in response cause a lot of the CNS symptoms. You can also have cancer associated with HIV infections. You have an increased incidence of malignancy. 40% of infected individuals develop cancers. One reason is because you don't have a functioning immune system anymore, and that's very important for surveillance and clearing tumors before they can get started. You don't have good immune surveillance. Another key, which we'll talk about next time when we talk about transformation and oncogenesis, you're making a lot of cytokines. You have huge levels of virus all the time. So your sentinel cells are always making cytokines. And one of the things that cytokines do is to stimulate cell division. And when cells divide uncontrollably, that is a recipe for cancer, because the more cells divide, the more mutations accumulate, and eventually the cell will become tumorigenic. And also, other viruses play into this as well. So uncontrolled replication, coupled with infection by other viruses besides HIV, also leads to tumor formation. So one example is Kaposi's sarcoma, a tumor of the connective tissue, which is shown here. Originally described in 1872, before AIDS, found mostly in elderly Mediterranean men, but occurs in 20% of HIV-infected homosexual men and 2% of HIV-infected women and transfusion recipients. So the incidence markedly higher once HIV is present. These develop, they require infection with a second virus, human herpes virus 8 or Kaposi's sarcoma virus. And in fact, these Mediterranean men that had Kaposi's sarcoma before AIDS era also had HHV8. So HHV8 will cause Kaposi's sarcoma, but people with AIDS are immunosuppressed, so they're more likely to be infected with both viruses. So that's one kind of tumor that is associated with AIDS. B-cell lymphomas are another type, higher prevalence in AIDS patients than the general population. These often result in tumors in lymph nodes, intestine, CNS, and liver. And we think that as the lymph nodes are destroyed, remember I showed you that progression of destruction of a lymph node, one of the consequences is that normal pathways to apoptosis, which regulate lymphocyte development in a lymph node, are destroyed. And so you get uncontrolled replication and the development of B-cell tumors. So these are often associated, again, with infection with either HHV8 or Epstein-Barr virus. And this is an example of what I've told you. We have HIV infection of macrophages and CD4 positive T cells. The cytokines that are produced stimulate the proliferation of B cells, endothelial cells, or epithelial cells. And we talked about how infection with HHV8 will then lead to Kaposi's sarcoma. So the combination of rapidly dividing cells and infection with HHV8, Kaposi's sarcoma. If these are B cells, rapid multiplication followed by EBV or HHV8 gives you a lymphoma, a tumor of B cells. And we didn't mention this, but even in epithelial cells, cervical epithelial cells, the same situation, rapid development, rapid division, human papillomavirus infection, carcinoma. So let's talk a little bit about an HIV vaccine. And this slide shows you one of the problems that we have, which we've already discussed. If you have this early acute phase of HIV infection where you have a lot of virus which is eventually cleared, it's cleared because of an immune response. You have a very nice antiviral immunity, and it's not cleared. The levels are reduced, but then you have persistence. So the one question is, how does the virus persist despite having good antibodies against it? And how does it eventually outstrip the immune control? And some of the questions for which we don't really have answers yet, and for which we do need answers before we can make a vaccine: do you need antibodies for protection and recovery? Do these have to be neutralizing antibodies? There's a lot of discussion now. Maybe just they don't have to neutralize infectivity. Maybe they could just bind to the virus and let it be taken up by other cell types. Do you need mucosal immunity to prevent infection? Or is immunity in the blood enough? And do you need T cells? We're not even sure about that. So here's an example of what HIV does with respect to the antibody response. So you get infected with a purple HIV here, and you make antibodies against it. You can see the purple antibodies here. But by the time these antibodies are produced, the virus has changed to an orange virus. So it's mutated, and these purple antibodies no longer bind to the virus. So your body detects this orange virus and starts to make orange antibodies. But by the time you do that, you now have blue HIV particles. And this is what goes on in that intervening period between the acute infection and AIDS. The virus is always outstripping the antibodies. It's sort of like Satchel Paige in a way, right? And here is a graph of an experiment which demonstrates this specifically. So we have a patient infected with HIV, and we take serum at month 0, 6, and 12. And then we react that sera with month 0 virus from that same patient and month 12 virus. Okay? So month 0 virus, by 6 months, you make a very nice immune response against it. Actually, you have a good immune response even at month 0. It gets stronger, and then it stays the same by 6 and 12 months. So 0 virus can be neutralized 6 and 12 months out. But the problem is that at 12 months, there is another virus which is not neutralized by the month 0 or month 6, and barely by the month 12 plasma. So this is a practical showing of how the virus is changing over time. So that's one of the problems. The virus changes so quickly that you can't neutralize it. Now, what would be the effect if at day 0 you had neutralizing antibodies? We don't know, because we don't have a vaccine that's been able to do that yet. And so we're not even sure if you block all infection whether the virus would still evolve to overcome it. Yeah.
Is it the speed of mutation, or let's say, because most of the viruses that we know have the error encapsulated at
Yeah.
stages, for
Right.
example, influenza virus
Right.
also changes very rapidly. But is it because HIV changes more rapidly than any other virus that we cannot have a proper antibody response in our virus?
Mm-hmm. No, so the question is, does this virus change more rapidly than any other RNA virus? So the error rate is the same. The problem here is that every day you make 10 times 10 to the 9th particles. So you have a huge productivity and turnover. So if you make one mistake per 10,000 bases, that's a huge number of mistakes. So every milliliter of blood has uncounted numbers of mutations. So it's a particular kind of disease here that lends it to this kind of escape. This is just another way of looking at these same data. These are, again, HIV escape. These are viruses taken at 0, 3, 6, etc., all the way out to 25 months. So these are from HIV-infected patients. And these are plasma taken at different times. And then you try and neutralize the virus with the different plasma. So every plasma is incubated with every virus sample to make this matrix. And then you mix the plasma with the virus, and then you ask, is the virus still infectious? That's a neutralization assay. So you can see the virus at 0 months is nicely neutralized by all of the sera from about day 3 or from about month 3 or so all the way out to 25 months. But the virus at 25 months, you can see, is poorly neutralized by the whole panel, because this has arisen at the point where this sample has been taken, and it's going to take another month or so to make antibodies, by which time you'll have a month, 26 virus. So, yes?
The numbers represent the number of antibodies?
Okay, sorry. So the number is the inverse of the dilution, where you get 50% neutralization. It's just a standardized way of calculating a neutralization titer. So here, 50% neutralization was at a dilution of 1 to 219. There's also evidence that CD8 positive cells, the so-called CTLs, are important for control of HIV. As the virus goes down, that's a period when you have an early CTL response. I showed a picture of that. If you do an infection of macaques with SIV, you can show that if you take away their CD8 cells, which you can do experimentally, they have a worse infection. So the CD8s are definitely contributing to protection. You also get selection of viral escape mutants. So CTLs recognize specific viral epitopes. The virus can change those sequences as well. Just like antibodies recognize an epitope, and those viral epitopes can change, the same thing can happen with CTL epitopes. They can change as well. So we see that during the course of an HIV infection, you get CTL epitope changes. So that suggests that they're important. Now here's a little bit more information on CTLs and elite controllers. So elite controllers fall into very specific human leukocyte antigen classes, B57 or B27. So you know the class 2 MHC molecules on your cell surface define how or participate in presentation of antigen to T cells. And there are many different kinds of major histocompatibility molecules in the population. It's highly polymorphic. And the more variation you have, the more foreign antigens you're able to recognize. And it turns out that these individuals with either a B57 or B27, you can have your HLA typed. If you're B57 or B27, you have a high likelihood of being an elite controller. That is, having infection but never getting AIDS. And what we think is that these individuals can accommodate even highly mutated epitopes in the MHC molecule. So remember, MHC presents a little viral peptide to a T cell. The T cell recognizes it as foreign and lyses that cell. If the viral epitope mutates, it won't be recognized by the MHC. But apparently these elite controllers have a MHC that can recognize a wide range of peptides. And it all has to do with the way the T cells are educated in the thymus. We don't have time to go into it, but it's a very interesting idea that very early in development, your thymus samples all kinds of peptides and decides which ones are going to be foreign and which ones are going to be yourself. So these kinds of HLA types apparently are very good at accommodating a wide range of viral epitopes. So here's an experiment where we take CTLs away from an animal. This is a macaque infected with SIV. So in a normal situation, you infect, they have a burst of virus, and then the CTLs peak and clear the virus at the same time. But if you take away the CTLs, which you can do experimentally, you get much more virus replication. So again, this is just suggesting that CTLs, in addition to antibody, are likely to be important for protection. So back to these elite HIV controllers. These are individuals that, as I said, can last for a number of years. 1 in 300 infected people are likely to be elite controllers, and they're associated with favorable HLA types, which I've already told you. There's no change in viruses that seem to be associated with this phenotype. They have virus their entire life, but they never progress to age. And if you want to listen to this, we talked about this on my podcast a number of months ago, where the idea is that the education of these T cells in the thymuses of these individuals is particularly different. So can you cure HIV infection? You can limit viral replication with antiviral therapy. But as I said, there are persistent reservoirs of infection. There are long-lived memory T cells, macrophages, even stem cells in your bone marrow or elsewhere that have proviral DNA in them. And that's going to be very difficult to eradicate. So I think prevention of infection is really going to be the key here. Unless we can figure out how to get proviruses out of these cells, we're going to have to focus on prevention of infection, and those individuals who are already infected are going to have to stay on antivirals for their entire lives.